welcome. Can you all hear me okay? Great, okay. So today we're going to be talking about algorithms for DNA data storage. I'm Ruthie Nachmany. I'm a software engineer based in New York City, and that's my Twitter handle. If you have any questions about DNA data storage, feel free to tweet at me. So in 2016, Microsoft set a new record for the amount of data written into DNA. They encoded and stored 35 distinct files, totaling 200 megabytes of data, using about 2 billion nucleotides, or DNA base pairs, in total. These files included This Too Shall Pass by OK Go, which we just watched a clip of, the Universal Declaration of Human Rights in over 100 languages, the top 100 books from Project Gutenberg, and a crop trust database of all the seeds stored in the Svalbard Global Seed Vault. And let's see, 1 second. And it was about this big. DNA is one of the most efficient forms of data storage we know of, which is important because we are producing so much data. 2.5 quintillion bytes of data are being created or captured each day. 90% of the data on the internet was created since 2016. And in addition to all the digital data we're producing, we're also starting to capture a lot of scientific data at a scale we've never done before. The Sloan Digital Sky Survey produces 73,000 gigabytes of data per year. The Large Hadron Collider generates 50 million gigabytes of data per year. In the next 10 years, researchers estimate that 40 billion gigabytes of genomic data are going to be stored, and we're going to store them for decades for regulatory reasons. And 1 billion people are predicted to have their genomes sequenced by 2025. So a lot of data. And we're starting to run out of ways to store that data. We're approaching the 1 terabyte per square inch capacity of hard disk drives. Past this point, temperature fluctuations can induce data corruption. We could use more heat-resistant material, but this would require major shifts in our storage infrastructure and a lot of associated costs. We also have challenges with current storage systems, like magnetic tape has an attrition rate of 22% and only lasts between 5 and 10 years. We have to keep rewriting data to it. And just to give you a sense of the scale we're talking about, the Large Hadron Collider that I talked about in the last example stores those 50 million gigabytes of data on a distributed grid of over 130,000 CPUs, 230 million gigabytes of magnetic tape storage, and 300 million gigabytes of online disk storage. So a lot of data, starting to run out of ways to store that. And DNA might be the solution. So DNA, what even? So some people refer to DNA as the code of life. It's basically, what gives our body instructions and what proteins to make, what genes to turn on and off. Just as a review, so there's 4 base pairs, adenine, thymine, cytosine, and guanine. A and T, adenine and thymine, bind to each other. C and G, cytosine and guanine, bind to each other. Some of the bonds are stronger, some are weaker. That's what causes the double helix shape. There's a phosphate backbone, and what you really need to know for the purposes of this talk is that there's 4 base pairs, A, T, C, and G, and if you're thinking about storing data in DNA, you're translating data in some way to those base pairs. So what makes DNA data storage so exciting? Density. DNA has an ultra-high storage density, as much as 100 trillion gigabytes per gram. It's 107 times denser than magnetic tape. Compactness, remember the image of the DNA and the tip of a pencil. It's an extremely compact form of data storage. Resilience, DNA doesn't have to be powered by an external energy source to retain data, as long as it's stored in a controlled environment. DNA can survive in less than ideal conditions for hundreds of thousands of years. We've recovered DNA from over 800,000 years ago. It also doesn't require active maintenance or rewriting every so often. Redundancy and replication are built-in features in DNA. Interoperability, obviously DNA has been around for a while, and it will continue to be around for a lot longer. So that's part of why we're really excited about DNA as a data storage mechanism. But it's also challenging for a variety of reasons. Cost, synthesis or creating and writing DNA, and sequencing or reading DNA is still expensive, and it's cost prohibitive compared to existing forms of data storage, like beyond kind of experiments and so on. The speed of reading and writing data to DNA is not yet comparable to digital storage. DNA sequencing can misread long strands of the same base, so that's a challenge that researchers have been working through. Data loss, data corruption. DNA, especially in a living organism, will mutate with insertions, deletions, and so on. A lot of these examples are using synthetic DNA, so there's less versus like DNA in a living cell, so it happens less often, but in all cases, there are going to be mutations or data lost with sequencing and rewriting, and with replication, these can get amplified. Corruption can also happen if the DNA is not stored correctly. Search, how can we find something specific without having to sequence all the DNA? Capacity, what's the maximum we can successfully write to DNA and have it be retrieved correctly? This is a question researchers have been exploring. The molecular structure, sequences containing a lot of the G nucleotide, for example, are difficult to write because they often produce secondary structures that interfere with synthesis. And so to solve all these problems, there's kind of requires an interdisciplinary approach that combines information theory, coding theory, computer science, compression algorithms, then also a deep understanding of molecular biology, synthetic biology, chemistry, physical computing, industrial engineering for the devices that are doing the sequencing and doing the reading and doing the writing of DNA, data to DNA. So it's kind of this really exciting interdisciplinary field, which is part of why I'm excited to share it with you today. So now we're going to talk about some of the milestones, how we got to where we are today and where the field is going next. So in 1977, the first DNA genome was sequenced, or read. It was sequenced in Frederick Sanger's lab. You can see a picture of him there. They sequenced the virus Phi-X175, a bacteriophage found in E. coli. How did we start to explore the idea that DNA could hold a message? In 1979, Japanese researchers Hiromitsu Yoko and Tairo Oshima asked whether the DNA sequenced by Sanger might hold a message encoded by extraterrestrial life. You can see an article from the New York Times at the time. Scientists examined tiny viruses for messages from outer space. It's a really fun read, and if you have a Time subscription, would recommend checking it out. And they weren't the first people to propose that either. In 1973, Dr. Francis Crick proposed the theory of directed panspermia, that alien civilizations may have put messages in DNA to disperse the seeds of life throughout the universe. So this was kind of exploring the idea, does DNA hold a message? Could DNA hold a message? The first time messages were stored in DNA was in 1988. You can see here a piece. It's called Microvenus. It was done by the artist Joe Davis in collaboration with the molecular biologist Dana Boyd. They took this ancient Germanic rune, that's in yellow, that represents life in the female Earth, and they encoded it into a bitmap image, you can see that over there, and then designed and synthesized that into a 18 base pair message and put that into E. coli, creating a living work of art. Since then, here are some of the things that have been stored in DNA. You can see a poster of Citizen Kane, smiley face emoji, computer operating system, early silent films, books about synthetic biology, Da Vinci, Mary Had a Little Lamb, I Have a Dream, Amazon gift cards, Claude Shannon's information theory, Muybridge's famous photography sequence, and obviously Bitcoin. So in 2001, it was the first example of naive encoding and translating. So what they did was basically translate each letter of the alphabet into a combination of DNA base pairs. A team of researchers at Mount Sinai School of Medicine wrote 2 Dickens quotes into DNA. It was the best of times, it was the worst of times. It was the age of wisdom, it was the age of foolishness. This totaled 70 bytes in DNA sequences. So basically they translated each letter of the alphabet as a combination of the bases A, C, and T. So AAA would be the letter A, AAC would be the letter B, and so on. So fast forward 8 years, so 2009, researchers introduced Huffman coding to kind of make storage of data in DNA more efficient. So researchers at the University of Toronto encoded more than 200 bytes of text, music, and an image from the nursery rhyme, Mary Had a Little Lamb, into DNA. You can see some of that here. To do this, they utilized Huffman coding, a lossless data compression algorithm where you basically assign variable length codes to input characters, and the lengths of the assigned codes are based on the frequencies of the corresponding characters. So the most frequent character gets the smallest code, and the least frequent character gets the largest code. This allowed them to utilize base assignment to enable efficient coding of characters. In 2012, researchers scaled up the amount of data written to DNA, and this kind of helped prove that storing data at scale in DNA could be very dense and accurate. So there are two main examples here. In August 2012, the journal Science published research by George Church, Sri Kasuri, and colleagues at Harvard University in which DNA was encoded with digital information that included an HTML draft of Church's 53,400-word book, Regenesis, How Synthetic Biology Will Reinvent Nature and Ourselves, 11 JPEG images, and this is my favorite part, a JavaScript program that displays DNA that can curve dynamically to follow the cursor. I looked really hard to find the JavaScript program, could not find it in any of their supporting materials, but I'm continuing to look, so follow up with me. In this work, they aim to illustrate that DNA, like digital media, can encode executable directives for digital machines. Multiple copies for redundancy were added. They developed a strategy that eliminated the need for long sequences, which are more likely to be misread by sequencers. Instead, they encoded data in distinct blocks and stored these in shorter, separate stretches, kind of analogous to storing data on a hard drive in discrete sectors. They encoded 1 bit for every base pair, so encoding A or C as 0 and G or T as 1, so about half of the theoretical maximum, which would be 2 bits. In total, the team made 54,898 oligonucleotides, or stretches of DNA, each including 96 base pairs of data, along with a 22-base sequence at each end to allow the fragments to be copied in parallel, and a unique 19-base address sequence marking the segment's position in the original document. The resulting DNA held about 650 kilobytes of data in such a compact form that the team predicted a storage potential for their method of more than 700 terabytes per cubic millimeter. So this result represented a significantly larger volume of data than had previously been artificially encoded in DNA, and it also showcased a data density for DNA several orders of magnitude greater than that of state-of-the-art electronic storage media at the time. So you can kind of see that here. Shortly after, in January 2013, Nature published research from Nick Goldman at the European Bioinformatics Institute, where over 5 million bits of data were stored, retrieved, and reproduced, including all 154 of Shakespeare's sonnets, included one here, a classic scientific paper, Molecular Structure of Nucleic Acids by James Watson and Francis Crick, a medium-resolution color photograph of the European Bioinformatics Institute, a 26-second excerpt from Martin Luther King's 1963 I Have a Dream speech, and a Huffman code used in this study to encode data. And importantly, all the DNA files reproduced the information at 100% accuracy. So the main innovations in this research were the use of an error-correcting encoding scheme to ensure the extremely low data loss rate, as well as the idea of encoding the data in a series of overlapping short chunks identifiable through a sequence-based indexing scheme. The sequences of the individual strands of DNA overlapped in such a way that each region of data was repeated 4 times to avoid errors. 2 of the 4 strands were constructed backwards, also with the goal of eliminating errors. To ensure that no base was used twice in a row, the digit that each DNA base encoded depended on the nucleotide that immediately preceded it. So for example, A, C, and G were assigned to represent 0, 1, and 2 at the nucleotide immediately after a T, but following an A, the digits 0, 1, and 2 were encoded as a CGT. So this strategy basically helped avoid the creation of any long stretches of the same base, so again, bad for sequencing, while still making use of DNA's 4-base potential. So in this work, the cost per megabytes were estimated at $12,400 to encode data, $220 for retrieval, and they predicted cost-competitive sub-50-year archival storage in DNA within a decade. In 2015, Reed-Solomon error codes were introduced into DNA. So this is kind of like a tried-and-true error correction method that is used across a lot of different mediums, and it was a big step forward when it was brought into DNA, and basically every research work since then utilizes this. So in February 2015, researchers from Zurich reported perfect retrieval of 83 kilobytes of data in DNA. So the innovation they did was adding redundancy via the Reed-Solomon error correction codes, which are used in consumer technology, data transmission, broadcast systems, and more. So basically provided a signal processing technique for repairing corrupted messages in DNA. In 2015, random access was introduced into DNA, which basically meant that instead of having to sequence all of the DNA and then translate it back, whatever algorithm you used to translate your data into DNA, translating it back, and then find what you wanted, you could search for that exact piece of information. So that's a major step forward in terms of getting to parity with other forms of storage systems. So in September 2015, a researcher named Olgika Milenkovic in her lab at the University of Illinois introduced random access and easily rewritable DNA-based storage architecture. This was based on DNA sequences with specialized address strings that could be used for selective information access. It overcame drawbacks of existing read-only methods that required decoding the whole file in order to read one DNA fragment. And as a proof of concept, they encoded parts of the Wikipedia pages of 6 universities in the US and selected and edited parts of the text written in DNA corresponding to 3 of these schools. And in February 2018, Microsoft built on this work and announced they'd achieved random access in large-scale DNA storage, so kind of at a much larger scale of data that had previously been done. They selectively amplified specific sequences from a DNA sample of more than 10 million oligonucleotides, or stretches of DNA. And they created a library of primers that were attached to each DNA sequence. The primers were used as targets to select desired snippets of DNA through random access. Before synthesizing the DNA containing data from a file, the researchers appended both ends of each DNA sequence with primer targets from the primer library. And in 2016, we talked about this earlier, but Microsoft achieved a record amount of data stored in DNA. So again, kind of moving from these theoretical ideas to proof of concept. And they also announced a goal of operationalizing DNA data storage by 2020. In 2017, the DNA fountain method was introduced, and it dramatically, enhanced the information density capacity of DNA. And so, yeah, it allowed DNA to reach 85% of its Shannon capacity. So the researchers who worked on this are named Yaniv Ehrlich and Dina Zielinski. They're at Columbia University and the New York Genome Center. And the method they developed, they call it DNA Fountain. It stores data at a density of 215 petabytes per gram of DNA. And just to give you a sense of that scale, 1 petabyte is equivalent to 13.3 years worth of HD video. The technique approached the theoretical maximum capacity, or Shannon capacity, of DNA storage. Shannon capacity is named after the information theorist Claude Shannon. It defines the notion of channel capacity, or the upper bound in which information can be reliably transmitted over a communication channel. Their encoding method achieved a 60% increase in storage capacity over previously reported efforts. And so basically the way they did this is they started with 2 basic principles. 1, not all DNA molecules are created equally, you don't want long stretches of the same base pair. 2, not all DNA molecules are going to make it. There's going to be noise and errors and so on, so handling that. And so their encoder works in 3 basic steps. First, it preprocesses a binary file into a series of non-overlapping segments of a certain length. Then they use this thing called the Luby transform, which is a relatively recent addition to coding theory that sets the basis for fountain codes, which are used in a variety of communication standards. And so what it does is it packages the data into short messages they call droplets, which are transmitted over a noisy channel. A user can recover a file by collecting a subset of droplets as long as the accumulated size of the droplets is slightly bigger than the size of the original file. The droplet contains 2 pieces of information, a data payload and a short fixed-length seed. The seed corresponds to the state of a random number generator, and it allows the decoder algorithm to infer the identities of the segments in the droplet. And then they screen the droplets, kind of make sure that it has, an ideal set of DNA sequence or base pair sequences that there won't be an issue with reading it or so on, or too many G's or whatever. So they screen the droplets, and if it doesn't pass the test, they just trash it and make another droplet. So it keeps iterating over droplet creation and screening until a sufficient number of droplets are created. And then, once they recover the droplet by mapping the read to its original binary format. They reject droplets with errors based on the Reed-Solomon codes, and they employ a message passing algorithm to reverse the Luby transform and obtain the original data. So beyond kind of proving that we can get to this really high level of information density in DNA, it can also reduce the amount of sequencing required for data retrieval. So one of the caveats of DNA data storage is that previous to DNA Fountain, each retrieval information would consume a portion of the material. And so while it's possible to copy the data, it also introduced noise. And so with this, you can do a deep copy, and it doesn't introduce the noise. So theoretically, you can copy data virtually unlimited number of times and preserve the data integrity in full. And so this is what people saw when they decoded the data. This is a movie called The Arrival of the Train at La Ciotat. It's one of the first silent films, and basically when people saw this movie in theaters, they ran away because they thought it was a real train coming towards them. There's a computer operating system, Da Vinci drawings, I think, and then Claude Shannon's information theory treatise. In 2017, DARPA started the Molecular Informatics program, which basically seeks a new paradigm for data storage, retrieval, and processing. Instead of relying on binary digital logic, von Neumann architecture, molecular informatics seeks to investigate and exploit the wide range of structural characteristics and properties of molecules to encode and manipulate data. And so to date, they've given out over 15.3 million in grants. So one of the big challenges with DNA data storage and kind of bringing it closer to parity with other forms of storage is cost. And so in 2017, we started to have cheaper sequencers called nanopore sequencers, but they're also much more error-prone, and they're error-prone in different ways than traditional, more expensive and more accurate sequencers. And so in 2017, the researcher who worked on the Random Access Project, Milenkovic, she published research that basically devised error-correcting algorithms specifically aligned with the kinds of errors that these sequencers make. You can see this is kind of what they wrote, Citizen Kane poster and the smiley face emoji. And then this is like what originally came back, and this after their error-correcting algorithms, you can see it was recovered fully. So they achieved the error tolerance via consensus sequence construction algorithms and asymmetric deletion correcting codes that were tailor-made for this kind of sequencer. And the consensus method they used combines multiple sequence alignment methods and side information provided by the address sequences of the DNA segments. And the deletion correcting codes they used were designed for errors that occur in these consensus sequences. In 2017 was a really exciting application for DNA data storage, recording biological interactions in DNA, or some people call it molecular ticker tape. So in July 2017, researchers at George Church's lab at Harvard published research exploring how DNA could function as a biological recording device by using CRISPR-Cas9. So CRISPR-Cas9 is a molecular tool that combines special proteins and RNA molecules to precisely cut and edit DNA. During a viral invasion, the proteins go out and grab a piece of the attacker's DNA, then they store those in clustered, regularly interspersed short palindromic syndromes, which is what CRISPR stands for. So usually, traditionally, in a cell, these are used to recognize and deactivate a virus next time it attacks, but people have been using this technique to kind of do a more precise form of gene editing, like choosing what you snip, choosing what you put in, and kind of using that on purpose. So anyway, Cas9 is the protein that does the cutting, and Cas1 and 2 proteins tell Cas9 where to do the cutting. And they don't just, insert viral DNA at random, they actually put it in the order in which it arrives. And so that can turn the genome into a temporal record of whatever happens in a cell. So you can see how they tested that out. They encoded this a GIF of a famous photography sequence of a horse galloping by the photographer Edward Muybridge into the genomes of a population of living E. coli bacteria. They were able to retrieve and reconstruct the images with 90% accuracy. Storing data in living cells is also an interesting innovation here. Most research stores data in synthetic DNA, which is more stable and reliable, but it's more unpredictable and more prone to mutations when you store data in living cells, but there's also some really exciting applications there. In 2018, image search was introduced into DNA. A team at the University of Washington, led by Luis Ceze, launched a social media campaign to collect 10,000 images to be stored in DNA. This is the site they launched, #MemoriesInDNA, so people could submit photos, and they aim to develop a search by image feature in DNA, making molecules carry out computer vision work to extract, identifying features like color, texture, and shape. So basically, if you think chemistry offers a palette of molecular diversity, like structure, size, charge and polarity that can be harnessed for information processing. And Seze's lab extracted visual features from the crowdsourced images and then mapped them onto DNA. So each photo might get tens of thousands of unique DNA segments, one encoding for a curve or vertical line or patch of blue. And then his lab introduced a coded query containing complementary DNA base pairs corresponding to these visual features. So each query sequence would get a special coding of magnetic nanoparticles that could indicate all the sequences that were a match, and only these sequences would be selected, extracted, and then translated back into visual images. You can see some of the images that were submitted here. In 2018, a company called Catalog announced this innovative thing called the library approach. And basically what they do is they use prefabricated strands of DNA to reduce the cost of writing DNA versus synthesizing DNA base pair by base pair. So generally, like, a lot of the cost savings in the last few years have been around sequencing DNA, but the costs of writing DNA are still quite prohibitive. So, like, for example, like, there's a synthesis company called Twist Bioscience that charges between 7 and 9 cents per base. So, you know, if you translate that into a single minute of high-quality stereo sounds, that's stored for about $100,000. So not really competitive today. But in 2016, to give you another example, in 2016, when Microsoft set a record by storing the 200 megabytes of data in DNA, they used 13,448,372 unique pieces of DNA. So comparing that to Catalog, what Catalog does is they generate large quantities of a few different DNA molecules, like 20 to 30 base pairs long. They design a machine that can stitch those fragments together in programmable ways. And instead of mapping one bit to one base pair, bits are arranged in multidimensional matrices, and sets of molecules represent their location in each matrix. So they don't have to synthesize new DNA for each new piece of stored information. Instead, they can kind of like remix the prefabricated DNA molecules. And so what the innovation there is separating the process of synthesizing DNA molecules from the process of encoding the data. And so it requires less synthesis and thus reduces costs. And on June 26th of this year, they announced that they use this system to store the novel A Hitchhiker's Guide to the Galaxy by Douglas Adams and the poem A Road Not Taken by Robert Frost in DNA. They plan to make it commercially available in 2019. So that's kind of like where we're at today. A little bit of what's next. Bitcoin cold storage in DNA. There's a company in August, it's called Carver. In August, they announced a way to store Bitcoin passwords in DNA as a form of cold storage, so keeping passcodes separate from internet connections. DNA cryptography. MPEG-G, which is a standard for efficiently storing genetic data. The DNA emoji, which should be coming to our phones very soon. Enzymatic synthesis, basically designing DNA that's specifically designed for storage versus like what DNA was originally designed for, which is more like biological functionality. There's also researchers that designed DNA with 6 base pairs instead of 4, which again like is an exploration into how can we store data more efficiently and compactly in DNA. Generally, like DNA is pretty much considered just for archival data storage today, but people are exploring new ways to use it for more frequent reads and writes. DNA computers, there's a whole field that looks at how you can put Boolean circuits in DNA or create semiconductors that use enzymatic reactions and so on. And so DNA data storage can be a partner in all of these projects. There's also hybrid electronic molecular design, which is this kind of interesting field that's looking at like what can electronics do best, what can molecules do best, and how can we build devices that combine the two. And you know, like if you think about when we talk about computing on the edge and the Internet of Things and Internet of Things devices, like we think about how much computing can we do on the device itself. And so there's kind of a whole field exploring this in biology as well. And so DNA data storage can be kind of a key part of that. So in conclusion, molecules are the next frontier when it comes to miniaturization and Moore's law. And the problems in this field are going to be solved in an interdisciplinary way by combining coding theory, information theory, molecular biology, synthetic biology, chemistry, physical computing, industrial engineering, and more. And so that's why it's kind of exciting to learn about it today. By looking at the world around us, we can learn a lot about efficient algorithms. We know there's a lot of challenges in data storage in general in the future, and DNA might offer a solution. So I encourage you to follow the field in the next few years. There's going to be a lot of interesting innovations, and if you're interested in learning more, feel free to come up to me afterwards.